Hi everybody. I thought I'd give a brief video to explain this poster I've got for the 2025 meetings of the Society of Vertebrate Paleontology in Birmingham. So here's the title: How Reliable Are Fossilizable Data in Phylogeny Reconstruction? And the basic problem is that fossils are this, as I wrote in the text, as you can see, a pale shadow of the animals they once were. And it's a very legitimate and indeed worrying question if during the course of decay the crucial information that one would need to place them accurately in the tree of life erodes away. And so here's a hypothetical example that something very close to this has been discussed repeatedly in the literature, that your anatomical data applied or given an optimality criterion, maybe those data tell you, well, here it belongs in this part of the tree, when in fact, if you had all the DNA among other parts, it might actually be up here. Now, how would we know? How would we test this? Well, my little section empirical test basically takes advantage of the fact that the tree of life for many groups, at least for mammals and birds, vertebrates generally, I would argue, is, we know more than we don't know, right? That no one argues anymore about, the possibility of a clade of birds and mammals to the exclusion of say, crocodilians or lizards. No one argues about the possibility of a hyrax and a horse clade to the exclusion of, say, elephants. There's a lot that's settled in the topology of the tree of life, and we could use that to our advantage. So here I have my little graphic showing you that's actually a Sengi and Elephantilus maiorus from near a place called Azamvelo, 100 kilometers or so east of Pretoria in South Africa. Here's a fossil jaw of an Apternidus specimen from out in the western interior of North America. And let's say for a moment, let's pretend that the only data we knew for Elephantilus was its lower jaw, and not even the entire lower jaw. Maybe, well, based on this particular specimen, you wouldn't have any information about, say, for example, the coronoid process, because that's broken off, right? You'd still use, of course, the real character states that an elephant shrew, a Sengi, has, in this case, Elephantilus maiorus, but the kinds of characters that would be available would be dictated by a given fossil template. And just to give you a bit more information along those lines, here is a hypothetical fossil template, which is what I'll call that, with these numbers representing discrete character states that you would enter into a phylogeny matrix. And let's say all those question marks represent parts of, the middle ear or the ankle or some bit that just isn't known for your given fossil template. And then you take that template and you take all these living species, anatomical data for living species, and you just copy wherever you get a question mark in the template, you put question marks in the living subject. That's another bit of jargon, I guess, for this presentation, living subjects. Now, again, this is the artificial fossil, this row here that's highlighted in boldface type. You're keeping the states that are, the characters that are known for the fossil template, but you're using the states from the real living subject, in this hypothetical example of a Sengi. You're also keeping the character states that are known from inapplicables, right? So that's represented by this little dash, and you'll notice the corresponding character down here is not a question mark. It's retaining what a Sengi actually has. And then, of course, with very few exceptions, once you get into the Pleistocene actually, DNA is pretty much unknown, right? This is one of these parts of a fossil that for the vast majority of the tree of life, the vast majority of animals with extinction dates that are prior to, say, 1 million or 2 years ago, we don't have their sequence data. Maybe someday we'll get proteomic data that's slowly trickling out. But for our purposes, the state of the art today is that you generally don't have anywhere near the amount of non-fossilizable data for these living, for fossils as you do for living species, of course. And so in this case, the examples that I'll tell you about, it's all question marks, right? Any genomic data that you may have had for a living subject in this exercise of artificial extinction that I'm telling you about turns into question marks. Okay, the other thing we need, as I alluded to a moment ago, is a well-corroborated tree. Now, there is, at levels above the species, for reproductively isolated high-level clades, there really is one tree of life. There's one bifurcating pattern, at least for animals, that represents the history of life. I mean, that's one of the great discoveries of evolutionary biology in the last century or a bit more than that. And so the specific examples I'm going to tell you about, one is using a big data set, a famous data set from O'Leary et al. 2013, for which I'm showing you the well-corroborated tree here. And then another example I'll get to in a minute is using a data set focusing mostly on sericid lipotyphlins, on shrews. But let's start with the well-corroborated tree using the O'Leary et al. Data set. Here you can see marsupials. They just sampled the two species. The placental mammals are represented by Atlantogenata and the two boreoeutherian clades, one of which is Euarchontoglires in green, the other of which is Laurasiatheria in blue. Now, this bifurcating structure of this well-corroborated tree is based on, I'll just zoom in a little bit, data that are independent from the anatomical matrix that O'Leary et al. Use, right? So the anatomical data of this 4,000, actually no, the hard tissue characters in O'Leary et al. Were about 3,660, I think. And none of those data had anything to do with the reconstructions undertaken by Tarver et al. In 2016, Esselstein et al. In 2017, Foley et al. 2023. Those are all genomic level analyses that support this largely bifurcating branching diagram that I'm defining for present purposes. And I think it's legitimate. You'll make your own conclusions on that. But this is the well-corroborated tree. You'll notice that it's not completely bifurcating. So there are some areas of ambiguity. For example, it's still unclear where bats fit within Laurasiatheria, right? There is some ambiguity about tree shrews. Are they closer to Cleries? Are they closer to primates and Dermopterans? I think there are people out there who have strong opinions in that. I'm sure there are. But for my purposes, I don't really want to stick my neck out. This is a fairly uncontroversial summary of what genomic data say, non-fossilizable say about the structure of the mammalian tree of life, right? Okay, and so in this exercise that I want to tell you about, sorry, I want to get that back so you can read this left column. What I'm doing now is deleting the non-fossilizable data, and by treating individual living subjects, I'm not actually putting any real fossils in this artificial extinction study. I'm using living subjects, deleting all their non-fossilizable data, and asking the simple question, do the fossilizable data that we have, say, for a bat, for Nycteris, or for a squirrel monkey, or for a human for that matter, or a lesser Malagasy hedgehog, right? It's not a hedgehog at all, it's a tenrec, right? Or for Dromaeosaurus, do the hard tissue fossilizable data place those species where they belong according to independent genomic data? And I'm going to do this one fossilizable, one artificial extinction event at a time, or multiple. So that's some of the methodology. I guess I can dive right into the results here. I think I need my image up here to do that, right? So this is a rather complicated figure, but I shall walk you through it. You'll notice on the y-axis we have percent congruence with the well-corroborated tree. And I should have mentioned there are lots of different ways you can measure congruence. Robinson folds, or shared splits, or Martin Smith, my colleague, has a really nice R package called Quartet. I mean, he's got a couple of them, but one of them is Quartet. You can use quartets as a means to measure congruence. For now, all I'm going to use is shared splits, and I'm going to represent that on my y-axis. Suffice it to say that the higher up you are on this y-axis, the more congruent you are with a well-corroborated tree. Now, this is starting out with the O'Leary et al. 3,660 fossilizable characters, so hard tissue, bones, and teeth, etc. And congruence with the well-corroborated tree is on the y. The fossil template completeness is on the x. And in their data set, there are lots of fossils in that data set, and some of them are really bad, known from, just a shade over 10% of these 3,600 characters, and some are reasonably complete, with characters from the ear, the postcranial skeleton, the dentition, the whole shebang. And it's subtle, but I want you to observe that as fossil template completeness goes up, so does congruence with the well-corroborated tree. Now, there seems to be an asymptote here, right? About 50, 55, 60%, you don't really increase anymore. So that's kind of an interesting result, and one that contrasts with the next data set I'll get to in a moment. The other thing I should point out is these solid red circles are using real character states for the various artificial fossils, for these subject taxa. You know, so a Sengi really, most species of Sengi, Elephantalis majorus, has 2 molars in each quadrant, not 3 as opposed to most placental mammals. And so, you know, you're representing that kind of information in these solid red circles. Whereas in these translucent red circles, the red circles that are somewhat translucent here, these represent randomized binary character states, right? So there's no phylogenetic data here. And, you know, if you're putting in random data, the paleontologist listening to this will be relieved to know that real character states of fossils do a better job at reconstructing at getting high congruence with the well-corroborated tree compared to randomized data. So, you know, it's kind of a low bar there, but nonetheless. But still, perhaps the most important point so far is that there is a subtle but nonetheless observable increase in congruence as the fossil templates get more and more complete, although, again, with the qualification that there's a bit of an asymptote there. Okay, so here the y-axis continues, right? So this dotted red box represents the bit that I've sort of snipped out on the left side, because, of course, I treated not just one artificial fossil at a time. You know, say Dromaeosaurus was the only one, and all the other several dozen extant subjects were kept in there with their real character states and all the genomic data, et cetera. That's why you get, you know, 70+% congruence with the well-corroborated tree using randomized characters, because those randomizations only apply to the one artificial fossil that I did per analysis. So I did 3 other analyses in which there are 15 artificial fossils, you know, so taking Dromaeosaurus and Erinaceus and, you know, Medoqua and Hippopotamus and all these different, 15 of these extant subjects, deleted their non-fossilizable data, just using the real character states present in a given fossil or present in a given extant subject based on the characters present in a given fossil template. I did 15 of those at the same time, then 25, then 35, and this is all color-coded here, right? So the pink salmon color represents 15 artificial fossils per analysis, the green represents 25, and the blue represents 35. Again, solid represents real character states, and these translucent regions represent randomized binary character states. And here, we're much farther down the y-axis of congruence. So here that's half of the nodes in the well-corroborated tree are recovered. But perhaps the most important result is this increase. As the fossil templates become more complete, congruence with the well-corroborated tree also increases. And this is evidence of statistical consistency, right? The more data you throw at a problem, the closer you get to the correct answer, right? And again, correct answer in this case is congruence with this. In O'Leary's case, O'Leary et al., it's this well-corroborated tree based on independent data, right? The branching structure here was not determined by the anatomical characters that are increasing in completeness over these various fossil templates. Again, with the O'Leary et al. Data set, there seems to be a bit of an asymptote. As you get around 60% complete, you don't really increase congruence. Now, the O'Leary et al. Data set from way back in 2013, that was a tough, that's a tough problem. Those are events that, in my opinion, certainly predated the Paleogene, Cretaceous-Paleogene boundary, namely the diversification, the origin certainly of placentalia, and the diversification of these major groups of placentals from each other, Afrotheres versus Laurasiatheres versus Urocondyglures. In my opinion, all that is pre-Paleogene boundary, pre-65 million years ago. Not necessarily by much, but I think it was almost certainly before then. And that's a tough problem. That's a harder problem than the next example I'm going to tell you about, which is the shrews, which obviously are placental mammals themselves. They predate all of the diversification of the high-level placental groups, things like Laurasiatheria, because of course shrews are within Laurasiatheria. So let me get now to the second data set that I examined for the Lipotyphla. And you're looking at this, well, I'm biased, of course. I think this is an absolutely stunning fossil, Domnina gradata, described just about a year ago by my colleague Robert Haubo Yuan and Quentin Martinez and Patrick Arnold and Ephraim Dixon and yours truly, published in Proceedings Royal Society B. Here's the QR code. Please excuse my salesmanship in encouraging all of you guys to navigate to that paper so you can learn about this really beautiful shrew fossil, because that's what it is. It's a heterocercid relative sister group to living shrews. And we did the same artificial extinction analysis here. We wanted to know, did the characters that we can infer from this, from Domnina, which is not just the skull, it's also represented by a really nice fossil hand and a bit of a forelimb, that the only association that's well preserved, at least, of postcrania with a decent skull, as you can see here, of any saurochoid, any shrew or total group shrew. So, okay, what are the results? Sorry, that's a bit of, I need to tell you a little bit about my poster here, other than brag about this wonderful shrew. Here's the well-corroborated tree, and this is based on non-fossilizable data that are independent of the anatomical characters that we use to place Domnina and other fossils in the tree of life. Same general idea, congruence measured in shared splits with the well-corroborated tree, this thing, right? You can see the shrews in red, the sauricines in red, the crocodurines in green, the brown colors, the talpid moles. Here we have some hedgehogs and moonrats, and then here is Selenodon and Isophonte. So Lipotyphla sensulatu. I guess I should point out that the eagle-eyed among you will see there are some polytomies here. It doesn't matter. You can still measure congruence with a largely resolved topology, well-corroborated topology that has a few polytomies. So there are some. Here are the results with just one artificial fossil at a time represented by AF1 in that red color, 2 artificial fossils at a time, green, 4, 8, 16, 32, as you can see. It stands to reason. It's easier for anatomy to get results that are congruent with the well-corroborated result when there are a lot of non-fossilizable data in that matrix, right? So if you're only making one artificial fossil at a time, only deleting, say, for Cryptotus parva, you're deleting its DNA alignment. You're deleting all the anatomical characters that are not known for, say, Domnina, which would include, for example, there's no ankle that I know of Domnina gradata. So you delete all that. And if that's the only artificial fossil you have, then you do that same thing with all these other extant species, and you still generally get pretty high congruence, ranging from, mid 80s to low 90% similarity. But of course, as you increase the number of artificial fossils, that level of congruence goes down. And what's more is that the sort of slope of that line, here on the x-axis, we have fossil template completeness, right? So our least complete fossil that we sampled in the Yuan et al. Data set from last year, Proceedings Royal Society B, the least complete fossil is this thing called Cercolestes, just barely over 10% complete. Baramendia is a fossil total group shrew. Actually, I think it's crown group shrew. Intermediate to that. We have, unfortunately, this large gap for reasons maybe one of you can explain to me. There aren't any fossils historically associated with Sauricidae that are between 40 and 68% complete, and that was largely because there weren't any complete fossils of shrews until we described at least shrews with reasonably well preserved postcrania. There's a Chinese fossil called Lusorex from the Miocene that's it's a whole animal but it's in a Lagerstadt condition, so it's really pressed flat. Gerhard Storch was, I think, the corresponding author on the paper that described that back in 2004, I think. So it's really hard to code characters from that because everything's really flat. But Domnina gradata, this University of Wyoming specimen in Yuan et al., does have 3-dimensional preservation of at least a forelimb, partial forelimb, and a nice skull. Anyways, what was my point? My point was that as these fossil templates increase in completeness, you get a really striking increase in congruence with the well-corroborated tree. There isn't that asymptote that the O'Leary et al. Data set had. Now, I'm not, this is an easier problem than the phylogenetic issue tackled by O'Leary et al. This is a relatively more recent evolutionary event, the divergence of, shrews from hedgehogs, from moles, from Solenodon, and indeed the divergences within Cericidae itself. So, I hope I don't come across as bragging and say that there's anything intrinsic about the Yuan et al. Data set that's better than O'Leary et al. It's certainly it's a lot smaller, right? So we had, 217 characters compared to 3,660. But it is different in the sense that there is no asymptote, right? You keep getting more and more complete fossils, and you're reaching towards 100% here, congruence with a well-corroborated tree. I mean, we don't know. That's an empirical question. Once someone does find a nice three-dimensionally preserved fossil shrew that's more complete than Domnina gridata, it's not hard to do, at least in terms of sampling. It's going to be hard to find that fossil, but it's not hard to be more complete than Domnina, because even with that forelimb, it's still missing lots of data. That's the general summary, I guess, of the second data set. And let me just conclude and say that this analysis supports what paleontologists have long assumed, but rarely tested, right? The poorly tested assumption that fossilizable data retain phylogenetic signal. They do, those fossilizable data. In the context of combined data analyses, your best bet is to always include molecular and anatomical data together whenever you can. We understand, of course, that fossils generally lack those molecular data, but to the extent that you have an anatomical data set for a fossil and a living taxon, and you have molecular data for that living taxon, that's going to help place the fossil accurately in the overall tree of life. And I think I'll end it there, other than one more point, I guess, which is that Martin Smith and I have a paper that touches on all of these subjects and others, although we didn't talk about the O'Leary et al. Or Yuan et al. Data set in that paper. We talked about several others, and here's the QR code for that will get you to the paper in systematic biology, whereas here's the citation for Asher and Smith, 2022. So with that, I shall thank you very much for your time.